Hello, everybody. No, it's not Mr. Anderson. It's Mr. Andrew Chung. And my partner Izzy and I are about to give you guys the scoop on evolution.
Going back to 1831, Darwin sailed on the HMS Beagle on an expedition around the world. He would make observations that would create the evolutionary theory.
Evolution is the process of how organisms of today have descended from previous ancestors.
Darwin was influenced by other scientists of his time, such as Hutton and Lyell, two geologists, Lamarck, who advanced a theory of inheritance of acquired traits, which was later proven false, and Malthus's economic theory that human population growth was limited by scarce resources.
Using his studies, Darwin observed a struggle for existence, where an organism's survival and ability to successfully reproduce offspring were entailed in one's fitness. Those who were more fit would continue to pass their genes on to the next generation. These genes coded for adaptations that better suited an organism. Thus, Darwin called this natural selection.
For instance, Darwin observed the finches of the Galapagos, and through study he noted that the finches were of different species that were descended from the same ancestor from mainland South America. Darwin noted that the finches had developed different anatomical structures, such as different beaks that would better suit themselves in their environments.
After Darwin's studies, Gregor Mendel studied how genes affected the successive generations of peas. Genes have various forms called alleles, which determine how the organism will develop and appear.
Mendel's studies showed how gene pools are vital to the evolutionary theory, since evolution is also any change in the relative frequency of alleles in a population.
Genetic variation can happen mainly in two ways. Mutations in genes are changes in the DNA sequence. Gene shuffling from sexual reproduction is another reason, which explains why one does not look exactly like one's biological parents.
Traits in an organism can either be controlled by a single gene or by multiple genes, which are called polygenic traits.
Polygenic traits are also affected by natural selection, in which the distributions of phenotypes can appear in any of three ways: directional, stabilizing, or disruptive selection. Directional is when individuals have higher fitness at one end of the bell curve than those in the middle or the other end. Stabilizing is when individuals in the middle have higher fitness than those at the ends of the bell curve. Disruptive is when individuals at the ends have higher fitness than those in the middle of the bell curve.
Genetic drift also can affect allele frequency when there is a random change to it. The founder effect is a particular problem where allele frequencies change because a small subgroup of a population migrates away from the main group, and the allele frequencies of the new population are severely reduced.
The Hardy-Weinberg principle shows how a population can keep its allele frequencies constant by controlling 5 factors from changing. That is, there is number 1, random mating; number 2, large populations; number 3, no migration or emigration of a population; number 4, no mutations; and number 5, no natural selection. If a population follows the Hardy-Weinberg principle, then it is in genetic equilibrium.
The forming of new species from changes in relative allele frequency of a population is called speciation. A new species is usually recognized when members of two populations cannot interbreed. Thus, they are reproductively isolated by 1. Behavioral, 2. Geographic, or 3. Temporal or timing reasons.
Scientists also use the fossil record, a body of information from fossils, to study evolution and its patterns in the history of Earth. Because fossils form in sedimentary rock over a great period of time, it preserves organisms that lived from long ago.
Thus, the deeper that a fossil is in the ground, then the older it is relative to the Earth's history. Scientists who study fossils are called paleontologists. Paleontologists use the geological timescale by studying rock layers and index fossils. There are several eras that mark different key fossils in the Earth: the Precambrian era, the Paleozoic era, the Mesozoic era, and the Cenozoic era.
Radioactive dating is the use of half-lives to determine the age of a sample, usually using carbon-14.
Earth's original atmosphere did not contain oxygen. Scientists Miller and Urey conducted an experiment to replicate the conditions of the original Earth's atmosphere, which contained the gases of hydrogen, carbon dioxide, water, and ammonium. Earth also originally had prokaryotes, which lacked a nucleus.
Then, eukaryotic cells appeared, which did have a nucleus. In cells, mitochondria, and chloroplasts, these structures actually contain their own DNA and ribosomes, and have membranes strikingly similar to prokaryotic organisms. Thus, the endosymbiotic theory suggests that mitochondria and chloroplasts were originally independent organisms that were consumed by bigger cells into a symbiotic relationship. As seen on Earth, a species may evolve through natural selection by changing into diverse forms that live in different ways. This characterization is called adaptive radiation.
Other patterns in evolution are also important to understanding how organisms evolve.
Convergent evolution is a description of how unrelated organisms may resemble each other, which is seen in both plants and animals. Coevolution is when two species evolve in response to changes in each other over time.
Scientists also postulate that evolution either happened through long periods of no change and abrupt periods of sudden change, called the punctuated equilibrium theory, or that evolution occurred slowly and steadily in a gradualistic equilibrium theory.
Developmental genes in body plans also contribute to evolutionary theories because scientists can look at fetal development of several species and compare them to their adult forms to their embryos.